Hi, Yadong.
Hi, Aaron. How are you?
Good. How are you doing?
I'm fine. Where are you? You're in San Francisco or
Shanghai.
Shanghai?
Shanghai.
Oh, good.
And you? San
Yeah, I'm
Francisco?
in actually I'm in Danville now, East Bay at home.
Oh, wow, it's fancy.
Mhm.
Nice. So you commute every day from Danville?
Sometime, for the working day, I usually live in I have a condo in the close by UCSF area. So just walk
Oh,
about 5 minutes.
okay. Nice. Do you want to test your slides?
Yeah, let's see whether I can share. Uh, mm, you see my slide? Let me see
Yep.
this
Yeah.
presentation.
Yep. Perfect. Yeah.
Okay,
Nice.
works. Thank you.
Yeah.
So I guess
When's
I'll
the last
go
time you?
second. I'm right.
Yeah, you'll go
Mhm.
second. When have you been back to China? Not in a while?
Oh, haven't been back about 2 years. The last time I
Okay.
was there was later 1990. I'm sorry, 2019,
Mm-hmm,
November
mm-hmm,
sometime.
mm-hmm.
And then I planned twice early this year, but finally just canceled. Just don't have that enough time. There's a 3 weeks,
Yeah.
quarantine.
Yeah. The quarantine's rough.
Mhm.
How's Gladstone doing?
Oh, it has been good. So at Gladstone, we have about almost 100% vaccination rate.
Oh,
So has been back to close to normal until recently this Omicron issue comes out. It's a little bit tightening now, the
hmm
control a little bit.
hmm 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 Um, hey, Juan, can you test your slide?
Sure. Yeah, let me do that. Okay. Hi, Aaron. Nice to meet you.
Hi, good to see you.
Mm, Yeah. Okay. So I'm gonna do. Okay. You see my slide?
Yeah, and can you do? Yeah,
Okay, that's good.
yeah, it looks good. You have a lot of books.
Yeah.
Cool. And what's that? There's like a what's that thing behind you? It's like, is that a
Bookshelf.
okay, okay, cool, nice. How's Beijing doing?
Beijing is doing great.
Good. Am I allowed to come to Beijing? I have a little star on my phone. Are we allowed to come to Beijing or not yet?
Yeah. But there is a lot of travel, right, policy now, right?
Yeah. I'm going this weekend to Xishuangbanna
Mm.
for a
From Banna.
little trip.
Wow.
Yeah,
Wow.
yeah.
Yeah. It's a good time to go to Xishuang Banna.
Yeah. We booked like a nice little villa in the woods where there's no one else. And I could just relax and read some books.
It's a nice place. I visited there almost 20 years ago.
Well, yeah. But I'm still a little bit nervous, just because travel plans can get canceled like in a day, like within a day's notice. So we booked
Right,
everything, but
exactly.
hopefully we can go, and then hopefully we can come back.
Mhm.
My friend, he was having coffee with someone in Shanghai, like in a outdoor coffee shop, and then it turned out the person he was having coffee with took a train a week or so ago, where someone else in the train, like a different car, tested positive for COVID. So then like that friend got like dragged out of his house in the middle of the night and had to quarantine for 2 weeks.
Mhm.
Because even if you're like a secondary contact.
Right. That's like a network. So you never know what will happen, right?
Yeah.
Mhm. Yeah. So
Yeah.
hopefully no one travels to Sichuan who gets
Right.
positive.
There are a lot of uncertainties. For example, the train you took or the plane you took, there might be some passenger. Mm,
Well, life is about uncertainties, right?
sure. So that's what I say. Yeah.
Yeah.
Aaron, how long you will be there?
You're asking the right questions. Right
He's
now
not
it's hard.
certain how long he will be there.
Yeah. Yeah, there's a lot of uncertainties in life.
Excellent.
Hi, Ron. Hi, Ron.
Yeah.
Hi, Huan.
Yeah.
Hi.
Hey,
Come up to apologize. So I'm in the early 9 a.m. meeting. So starting already. Okay, then that's at 8:30. So I'm sorry to miss the talk of yours, but I'm sure you can have great talks. Sorry. So there's
Yeah.
some meeting appointment that haven't couldn't move. Is that Yadong here yet?
yeah, Yadong's here.
Yeah.
Great.
Hi,
And
Yadong.
great.
Hey.
Yeah, looking forward. Looking forward to talk. I apologize for having to come to the state team lab meeting, the annual meeting. So some appointment
Mhm.
couldn't
No
skip.
problem
So
at all.
I'm sure people will enjoy your talks. Great. Okay, thanks so much for coming to your Zoom. Great.
Thank you.
Here.
Thanks. So long.
I see you. Bye.
I'm surprised they scheduled a meeting at the same time as NeuroZoom, but you never know.
But I know many people are preparing the defense for the brain project these days. I believe the
Okay.
defense will become from today the whole week. So there are a lot of colleagues or
Mhm.
other people in China who is busy doing this, especially
Okay.
during
But
this
you
week.
already have the Brain Project grant, right?
No, of course not.
Oh.
So
Oh.
yeah,
Okay.
I'm part of one
But
project, but yeah, but I'm not the person who presented. We're just
I see.
preparing
I
the
see.
slides.
Okay. Okay. Yeah. Yeah, things move really fast in China. Like someone normally someone invites me to give a seminar, I don't know, a year in advance, 6 months. The other day, someone texted me at like 9 p.m. said, "Can you come give a talk tomorrow morning?" So it was like it
Yeah.
was super short notice. Yeah. Okay. Looks like it's just me then. Okay. So are you guys ready? So why don't we get started? Thanks everyone for coming to another week of NeuroZoom. Really looking forward to uh, Huan and Yadong's talks. Before we get started with the talks now, let me advertise the next week's talks. These are going to be the last talks of the calendar year, and then we'll have a break for Christmas. But we have two really amazing talks coming. We have Hu Zhao and Guo Changbi, and sort of homework to prepare for next week is go read their papers, especially Hu Zhao's new preprint, where he presents a new amazing new clearing technique he's a dentist, so he knows how to embed and clear bones and things, and you just really amazing pictures. And then on the other hand, Guangchang has developed cryo ET technologies where you can actually see receptors on the membranes of neurons in full detail. So these talks are going to be visually amazing. Okay, so so please tune in then. Continue to send me or Zilong a note if you'd like to present your work. We have lots of great speakers in next year. So it's my great pleasure to introduce our first speaker, Dr. Huan Luo, who is a professor in the Department of Psychology and the investigator in the IDG McGovern Institute for Brain Research at Peking University. So she earned her undergraduate degree at Wuhan University, where she majored in engineering, and then did a PhD at the University of Maryland, College Park in cognitive neuroscience. And most of us can't say this, but in grad school, she solved an important problem in neuroscience. And she had many important papers, and her thesis research was to investigate how natural speech is represented in the auditory cortex. And she used really elegant magnetoencephalography recordings for participants that are listening to spoken sentences. And she discovered a certain temporal window in which this speech is segmented. And it sort of led to the idea that of the syllables as being discrete units for representing spoken language. She then continued to work on how not just auditory stimuli are processed, but how, let's say you're watching a movie that has auditory and visual stimuli, and how those are integrated together. And then that was an important paper in PLOS Biology, and the previous one was in Neuron. She then started her own group at the Institute of Biophysics in Beijing, and then moved to Peking University, where she is in 2015. And her lab continues to try to gain insights into how the brain temporally organizes sensory information. And she uses a combination of brain recording and mapping techniques. And looking forward to hearing her latest. So please take it away.
Thank you, Aaron, for the very nice introduction. Yeah, so am I starting now?
Yes, please.
Okay, thank you. Yeah, first, thank you. Thank Aaron for the invitation and also the excellent introduction. And I'm very excited to be part of this Neuro Zoom series. Today, I'm going to talk about something different. My lab has recently done, as shown in this title, where I'm trying to present some sequence working memory work in recent, my lab has done in recent years. Okay, so we know that working memory is a core ability in our daily life. If we make a simple analogy about working memory, that's like a post-it in the brain, which could temporarily maintain information in our brain for future decision or action. And so, for example, we can easily memorize a lot of color squares or a lot of digits, or even a pretty image like our PKU campus. And remember that a very typical experience in our daily life is sequence. For example, if you want to make a phone call to your friend, not only do you need to memorize 10 digits, but also their ordinal relationship. Otherwise, you cannot reach your friend. The similar principle applies to a melody. If you want to recall your favorite melody, you need to memorize a lot of pitches as well as the ordinal relationship in the sequence. Similarly, you can memorize a sequence of visual items or even a sequence of gesture movement. And today, as also introduced in Aaron's work, my lab is especially interested in the time-resolved environments of human behavior and also our human brain activities. The reason is that we know that dynamic inputs enters our brain to be processed, to be attended, and to be memorized. And also we know that information will communicate among different brain regions also in a very dynamic way. Sometimes as a type of neuron oscillations. This is kind of a topic my lab has previously done a lot. And importantly, we also know that, excuse me, neuron plasticity also occurs within certain temporal windows. For example, we know that it will occur around 40 to 80 milliseconds. So it's a very short temporal window for our brain to implement the neuron plasticity principle. Therefore, tens to hundreds of milliseconds, it's a very crucial temporal window in the brain to process and to store information. In other words, if we try to understand the neural mechanism for working memory, we should use a very time-resolved approach to look at the behavior and our brain activities. In fact, my lab's recent work have demonstrated some evidence supporting this idea, which I'm not going to get into details. For example, we show that attention is also a dynamic process and fluctuate between different locations, different features in a very rhythmic way, around 200 milliseconds. A similar idea comes to the working memory. We find some temporally compressed reactivation also in the working memory. Today, I'm going to present two parts. The first part is how a sequence of items and their ordinal relationship maintained in the human brain. In other words, how can we probe the sequence working memory? Second one is, could we modify sequence working memory in humans? That is, how could we manipulate sequence working memory? I want to make 2 points. The first one is that we do system level measurements. So it's not single cell recording, it's not intracranial, but non-invasive measurements in humans. Here, particularly MEG and EEG. Second one is we will focus on dynamic perspective. So we will be very interested to see the tens to hundreds of millisecond temporal scale. Let's first look at a typical working memory trial, which will consist of 3 stages. The encoding period, where subjects are presented with a sequence of items to be memorized, and then followed by the maintaining period, where they need to hold those information temporarily in their working memory. And then finally, recalling period that they need to be tested on the memorized information. And we are very interested in this maintaining period. And remember that during maintaining period, information is somehow represented in the brain, while there is no outside input. So this is a very interesting part. So how is information encoded or represented during this maintaining period? I'm just talking about recent evidence. Some recent evidence actually began to challenge the previous view and think that the information is silently retained in working memory. So for example, as indicated by this box, there is no sustained firing during this delay period. There is also no information representation during this period, although we know that information should somehow be retained there, right? So this is called activity silent working memory state. And some theories or some framework has begun to suggest it that instead of relying on sustained firing, the information is actually stored in the synaptic weights within this network. That's why it doesn't need to show sustained firing, because the information is just silently stored in this synaptic affixes. Then a natural question would be, if information is silently stored in the network, how could we read out information from working memory? And recent studies has begun to develop a very intriguing approach, which called impulse response approach. The basic idea is that you can just send a transient impulse to perturb the system. So the information that is silently stored in the network will be reactivated. I'm gonna to borrow a clip from Tom and Jerry to illustrate this idea. Okay, so for example, we know that Jerry is kind of like the information that is silently stored in this network. And that's the information that we try to detect from the system. So how could we detect? Tom could just send a huge pound to the piano, right? And this huge pound is like an impulse. And this impulse will reactivate Jerry from this piano. So this is a basic idea of the impulse response approach. Okay, so far so clear. Now let's look at our boring experiment. And this is an MEG experiment. And it's mainly done by my PhD students, Huang Li. And in each trial, human subjects are presented with 2 greetings, and they need to memorize the 2 orientation features as well as a temporal order. So now the orientation feature is like Jerry that we should memorize and that we should look for in the brain activities. And we know that after the human brain encodes the two orientation features, the brain will enter into activity silent state, which means you cannot easily decode or access the working memory information during this state. But then during this state, we present a white circle. This white circle is like a neutral pin to transient to perturb the working memory system. So the white circle is like Tom here. And our hypothesis would be whether just presenting a white circle Tom here will reactivate Jerry, who is silently hidden in this working memory system. So this is a whole idea. And I'm just cut to the chase and show you the data. And by using an inverted encoding model, we could actually decode our orientation information from the brain signals non-invasively. And as shown here, yes, indeed, we observed that this impulse could reactivate to-be memorized orientation features, right? The second orientation and followed by the first orientation. And I want to make 2 additional points. First one is that we showed a backward reactivation. Sorry, that's a reactivation. Which means the second orientation will be reactivated prior to the first one. And another thing is very crucial is that we see that it occurs at a very fast temporal scale. So you see, after the ping, around 200 milliseconds, the reactivation occurs. And also the time interval between the second and the first orientation features is around 200 milliseconds. So all the things comes at a very fast temporal scale. But at least it means that we could probe the working memory. And moreover, we show that this backward reactivation could predict our subsequent memory behavior. We're using the same trick also to auditory sequence working memory. This is also done by my PhD students, Fan and Han Qilin. What we did is ask the subjects to memorize 3 pure tones. So they need to memorize the pitch of the 3 tones as well as a temporal order. And so now Jerry will become the pitch of the auditory tones. And then again, after the information enter into this activity silent state, now we present a white noise. So now the white noises will not be the big white circle showing previous study, but just a white noise ping, like this type of sound. And we believe that this white noise ping will transiently perturb the auditory cortex while the pitch information is presumably maintained. And so the white ping will be Tom here. And again, we show that this white noise will also reactivate the memory pitch information from the human brain and is related to subsequent memory behavior. So the corrected trials will show larger pitch reactivation compared to the wrong trials. Okay, so far I have presented our work. So we're using an impulse response approach to try to use a neutral ping to transiently perturb the working memory system so that the memorized information will be reactivated. We're using a similar ideal to visual sequence and auditory sequence. And we show that this reactivation occurred at very fast temporal scale, tens to hundreds of milliseconds. And this reactivation is related to memory behavior. The second part, I think is the most exciting part, is that could we modify sequence working memory? So manipulate sequence working memory. This is done by my PhD student, Jia Qili and Huang Xiaoli. Let me summarize a little bit. So previous one, we just using the impulse response approach to passively detect Jerry. By using passive detection, I mean, for example, Jerry is there and we reactivated Jerry. Jerry is still Jerry. Jerry cannot be manipulated. How could we manipulate Jerry from this piano? What we should do is first we need to reactivate Jerry and then we do the manipulation of the Jerry. So in other words, we should first reactivate the memory information and then doing the manipulation. This is ideal. And this is also the hypothesis behind the dynamic perturbation we developed. Let me explain a little bit how we do that. In each trial, subjects still are presented with 2 bars and they need to memorize their orientation features. So orientation 1 and orientation 2. And then the information will come into activity silent state. And then during this recording period, the subjects need to rotate this horizontal bar to either the first or the second orientation, depending on the instruction. And we know that typical memory behavior for such sequence will be a recency fact, means the second orientation will be better than the first one, okay? This is called recency fact. And then how could we manipulate? We try to manipulate this activity silent state by applying a very time-resolved perturbations so that this recency will be modulated. This is the whole idea. What we did is presenting 2 flickering color probes. And the reason we use a flickering color probes is because we think the flickering will reactivate the working memory information. Remember, we should first reactivate it and then manipulate it, okay? And these 2 flickering color probes, the luminance sequences of these 2 color probes will follow 2 white noise, completely white noises that is randomly generated in each trial. So it's totally randomly generated. We call one is sequence 1 and one is sequence 2, which I'm not going to get into detail, but just remember 2 white noises. One is called sequence 1 and one is called sequence 2. And we try to introduce some temporal correlations between the sequence 1 and sequence 2 to modulate the item's relative memory strength. So the idea is that, okay, we reactivate the information using these white noises. How about we introducing some temporal correlations between them? And if we introduce additional relationship between them, will that affect the relative memory strength of these 2 orientation features? So that's the idea. First, let's look at the baseline. During the baseline conditions, these 2 luminance sequences will be independent of each other, which means there is no temporal correlation between them. And so there should not be any modulation for the memory, right? So this is just a luminance sequences and flickering independent of each other. And our results also show that, yes, such independent perturbation cannot affect the typical recency fact. So it's still a typical recency fact. Second item is better than the first one, okay? Now we introducing a temporal manipulation. The first one we do is called a synchronization condition. We just equate sequence 1 and sequence 2. So now sequence 1 is exactly the same as sequence 2. And I still use a Tom and Jerry clip. Now Tom and Jerry play the drum synchronously. That's what we mean by synchronization. And therefore sequence 1 and sequence 2 will share the same white noises in each trial. And our results show that just by presenting the synchronized 2 color, flickering color probes, the previous recency fact, okay, will be disrupted. Now there's no difference between the first and second one. So this suggests that temporal synchronization will disrupt recency fact. And the second one is we call auto reversal manipulation. What we did is we still randomly generate sequence 1, and then we shift the sequence 1 by 200 milliseconds. So now sequence 1 always leads sequence 2 by 200 milliseconds. Let me still give you a demo because this one is very abstract. So like Tom is always chasing Jerry and Tom is always lagging Jerry by 200 milliseconds in different contexts. That's like here. So sequence 1 will be Jerry, who is always leading sequence 2, Tom, okay? And we can do the same trick by reversing their role. So now sequence 1 will be Tom and sequence 2 will be Jerry. So now sequence 1 lags sequence 2 by 200 milliseconds. You might think this, let me show just giving you an intuitive way. And the above panel will red leads blue by 200 milliseconds. The bottom panel is red lags blue by 200 milliseconds. I don't think you can tell any difference between these 2 conditions because we know that this is beyond our humans' perception, okay? So remember, you need to memorize 2 orientation features. In sometimes I show you this one, sometimes I show you this one. Will that affect your memory performance? And yes, it did. So for the same order condition, we still see the recency effect, but for the reverse order condition, although you cannot perceptually discriminate it, it will reverse your recency effect. Now the item 1 will be better than item 2. Okay, we also build a model, continuous track network, which I'm not going to get into detail through collaboration, and fully replicates our results. And let me summarize this part. So for the baseline condition, when the 2 probes are independent of each other, we see the classical recency effect. And when you synchronize these 2 flickering color probes, the recency will be disrupted. And when you do a reverse auto, so changing the order of them, the recency factor will also be flipped. Now recency becomes primacy. So this is a conclusion of the two projects I have just mentioned. In the first one, probing sequence working memory, we use an impulse response approach to reactivate the maintained information from this activity silent retention stage. And this reactivation is related to memory behavior and occurs at tens to hundreds of milliseconds. In the second one, we developed novel dynamic perturbation approach, also in a very fast temporal scale to manipulate working memory. So, okay, this is what I'm going to show today. And I want to thank my lab members, very talented young students, my collaborators, my grant supporter, and also thank you for your attention. Thank you.
Thank you very much, Huan. We have
thank you.
Time for questions.
Should I stop sharing my screen or not?
Sure.
Okay. Yeah, this is, I know this is a very different field.
So what are sort of the next steps that that are the next challenges that you want to work on?
I'm still very amazed by working memory, because I think working memory is a process that is kind of independent of outside inputs. It's some kind of internal operation your brain do to memorize things, to manipulate things. For example, we know that our memory is not always true, right? We have added many our own experience and our own knowledge and even our own imaginations to change what we have really experienced. So I'm very amazed by how the brain does that and by using what type of way to do that. And the dynamic perturbation I have just mentioned in the second part is kind of in this direction. Because, you know, like in human subjects, we don't have optogenetics to precisely manipulate these things in a spatial temporal precise way. But here I want to show that even by presenting subjects with those flickering color probes, that has a lot of high detailed temporal information that could somehow affect your memory performance. So that gave us a way, at least for the healthy human subjects, to try to modulate their behavior. So that's some part I'm quite interested.
Do you have any interest or availability to test this in disease situation, subjects that have memory impairment or other language processing or visual processing defects?
We haven't done on any disease because I think first we need to establish the very stabilized paradigm on human subjects, and then we could try that. But I have a very crazy idea. For example, we know that, for example, cat is chasing dog. That's a sentence. But can you present something like unconsciously to subjects, and that will make your perception shift to dog is chasing carry, for example. So you totally reverse the relationship or the sequence of this thing. I think that's right. That's a very challenging question, but it amazed me a lot. Yeah. I see a question from this chat box. Could you please explain in just two? Okay. So I think if you imagine how do you maintain information during the working memory, classical work will show that there will be some sustained firing, which means the neurons that encode the to be memorized information will keep firing in a very sustained way to kind of represent the information, holding the information. But recent some evidence began to challenge this view because they show that it's not sustained, right? But still, animal or human still can memorize those information even without those sustained firing. So they propose that the information will be stored in the synaptic efficacy. The way that it could work is because we know that there will be some nonlinear dynamics for the synaptic efficacies. And so our single neurons they are not kind of passive detection. When information comes and then decays, the information will still be stored in the synaptic affixes for some time, right? So when it is still retained in the synaptic affixes, then that means that the memory could be stored there for a little bit. So that is what I mean by active silent state. So you cannot detect the fairing, but those neurotransmitters is still there and echo the previous history. And that's why we need using some way to detect the information from this silent state. I hope,
So for some of your auditory processing work, is that robust to different languages? Subjects hearing in English or Chinese, or what about subjects who are double language speakers? And
it's quite robust.
I know I really struggle processing Chinese that I'm hearing versus English.
Right. So that's my PhD work. So it showed that 200 milliseconds is kind of very crucial temporal window for the brain to segment the incoming natural speech. And that is quite generalizable across languages, because we believe that this temporal scale will be bound to the syllable length across languages. And we also know that syllable length across language is relatively constant. People have done a study trying to study the statistic properties across different language. And for music, it varies widely, but for language, I think the 200 milliseconds actually is from 150 to 250 milliseconds. It's quite a stable scale. So which means all the language will come kind of doing this fluctuation in this way. And the reason some people think is because of our production organ. So that's how we produce our language. And that's why this temporal window is so crucial. Right? You already know that any language will be like that. So your brain will just initiate this temporal segmentation, like a pseudo clock. It's not exactly clock, very strict clock, but just a way, pulsatile way to process incoming information. Right? But you will not use that window for, like, for example, white noise, because white noise is not speech.
Hey, Huan, that was great talk. A quick question. All this process will be affected by aging. If you do, at a different age of those tests, you will see different.
Right, that's great. Yeah, so I haven't tried the aging group, and some colleagues in China, they saying that maybe they should use this paradigm in aging group or Alzheimer's disease. We know also they have some working memory problems there. So yeah, that's our future direction. See whether we could establish a very short and easy to test a paradigm, and we can try that on different population or disease. Thank you.
Okay,
thanks so much, Huan. Great talk. And now we'll move to our next speaker. So it is a great privilege to introduce Professor Yadong Huang. Yadong is the professor at UC San Francisco, one of the best universities in Northern California. He's the director for the Center for Translational Advancement at Gladstone Institutes. And he received an MD from Qingdao Medical University and a PhD from Peking Union Medical College and the Chinese Academy of Medical Sciences in Beijing. He then did postdoctoral training in Germany, and then joined Gladstone Institute as a postdoctoral fellow, and then became a staff research investigator, and eventually was promoted. Now he's a senior investigator. Yadong is famous for his pioneering studies on mechanisms of Alzheimer's disease, and studying with a focus on the ApoE4, a genetic risk factor for the major genetic risk factor for Alzheimer's disease. He's provided compelling evidence that ApoE4 produces a toxic gain of function effect And he's shown this elegantly in iPS-derived neurons, in which he can reverse those effects by switching ApoE4 using gene editing back to ApoE3. And but more excitingly, he's developed a small molecule that has this remarkable ability to bind to ApoE4 and to change the conformation in a way to make it more like ApoE3, and this ameliorates detrimental effects seen from ApoE4. So this is inspirational because it shows that the basic research he's been doing in his laboratory for many years can now be translated into a therapeutic strategy for Alzheimer's disease. So it's great to have Yadong present his latest work and insights to us. Please go ahead.
Thank you so much, Aaron, for this quite very nice introduction. It's a great pleasure today to share with you our more recent work about precision medicine and drug repurposing for Alzheimer's disease. Let's see. So as all you know, this Alzheimer's disease is a multifactorial disease. It's probably we should call it a diseases or syndrome. It cover many different subtypes probably. Listed here are three major contributing factors, APP or A beta, ApoE4, and tau. All those three major factors probably in concert together with many other genetic or epigenetic factors or aging, injury, environmental factors lead to some neuropathology, neuroinflammation, and neurodegeneration, which can lead, of course, to neuronal network deficit, finally, to cognitive impairment. So my lab, as Aaron mentioned, has been working on this particular protein, ApoE4, try to understand how does this protein mutation cause or contributing to Alzheimer's disease. So if you read those literature about these three major contributing factors, you will see, A beta acts through multiple, molecules, pathways lead, to Alzheimer's disease. E4 goes the same way, and also tau. And plus, all these three can interact with each other to make a really complicated network of picture difference. So you can imagine if we target one of these two, this is only three, there are many more than this, for drug development, whether this would work. I would argue this may not work well because you take away one effect, there are many other effects still there. So what we try to see whether we can take some lesson learned from cancer field or infectious disease field to see if we do this new concept for drug development, so-called network concept of drug target. That means we will not target directly on this individual protein or molecule, but we move the target downstream to try to block the whole network change, so-called network medicine development. And in this case, what we try to do today, I want to share with you. So use this targeting ApoE4 genotype specific transcriptomic signature of AD as this network target, and in combination with computational drug repurposing to see whether we can really quickly identify some potential drugs for Alzheimer's disease treatment. So I would say this project has been leading by a very talented PhD student, Alice Tobis, in my lab. Some part of the work was in collaboration with my colleague, Marina Sirota at UCSF with her lab. So just a few words, why doing drug repurposing and precision medicine? As many of you know, the major issues for new drug development include a high cost, take a long time, and high failure rate. It takes roughly 15 years and over at least 800 million on average to develop and bring a new drug to market. So usually over 90% of drug candidates fail in early development. And one reason is because of heterogeneity of Alzheimer's disease that increase the failure rate of developing new drugs. So there are many discussions in the field about what will be the solution. So I believe one of the solution is drug repurposing and precision medicine. So drug repurposing, because it's identify the drug from existing approved drug, it lowers the cost. It also requires shortens short time for a drug to reach patient if it success. So if you combination with the precision medicine will further reduce the risk of failure on that. So for this, we'll start to do the first step is identify ApoE genotype specific transcriptomic signature of Alzheimer's disease. So we took one of the biggest public available database when we started this project, at least, that's from this Miami University, a transcriptomic data set of human temporal cortex, including 116 controls and 97 ADs. The breakdown for ApoE genotype for the control group, as shown on the left, is very classical, about 2% ApoE44 homozygous, about 20 to 23% E4 heterozygous, and the remaining about 75% are E33 homozygous. So if you look on the right side, this is also classical AD group. It's about 18% for ApoE4 homozygous, and another 50% about ApoE43 heterozygous, and 30% of E33. So it's a dramatically increased ApoE4 carriers in the AD group. So we stratify based on their ApoE genotype into E33, 34, E44, and then we compare E33AD to E33 non-AD, E44AD to E44 non-AD. So basically only compare within each genotype group to identify those so-called differentially expressed genes. As you can appreciate, there are about 1,000 upregulated genes and 1,000 downregulated genes altogether. So a few hundred about E44 AD in red, about 500 or 600, 500 about E33 AD to E33 non-AD. So the most striking figure here is that if you compare across group, quite striking to us, it was only about 6% the upregulated or downregulated genes are shared among 3 different AD subgroups with different ApoE genotype. That tells us we might truly need a different drug to treat those different subgroups with different ApoE genotype. So after we got this ApoE genotype specific AD transcriptomic signature, we apply those signature to query for known drug database covering a few thousand approved drugs. So for each drug, its transcriptomic perturbation profile is compared to ApoE genotype specific ApoE AD transcriptomic signature. So as you can imagine, this, let's say if it's E44 AD signature, when you compare to the different drugs, you'll find some drug may make it, similar transcriptomic change, but the other drugs maybe, go opposite direction. Those are the drugs you want to get. And those positive score drug will make it worse potentially. It could be useful for disease mechanism study. So you get a drug, get different scores. So the -1 is the best score you want to get ideally, and the positive one is the worst score. And then the hypothesis, the compound that reverses the AD transcriptomic structure specific ApoE genotype will potentially be therapeutic for that specific ApoE genotype subgroup of AD. So this, shown this slide, is the rank of few thousand compound against the ApoE44 specific AD signature. You can see the rank very differently. We look at the top 5 compound, and then we decide to focus on one drug called bumetanide, a so-called water pill. I'll get on that a little bit later. So this the number 4 drugs. We did not take the first 3 because some cancer drug is really toxic to cells sometimes, and another one is antibiotics you cannot use for long-term treatment for Alzheimer's disease. And if you look on the right side, those are those genes altered in E44 AD. If you look on the right side, this bumetanide treatment on cells will reverse many downregulated gene to up direction, and some upregulated gene went down. So this is quite encouraging to us. And then we also use the same database query for E3-4AD transcriptomic signature or 3-3, you see that each one generate different drug list. Bumetanide was still among those negatively scored compounds, suggesting they might work, but they are not the best one. Those are usually around about 200 to 500 positions. So we decided to focus on the bumetanide potential on the E4-4AD. So just a few words about this drug. Is the brand name called Bumax or Buranex. So it's a loop diuretic that have been used in clinic for more than 40 years. It's often used to treat edema or in heart failure, sometimes also as an anti-hypertension drug. It's originally developed as an inhibitor for NKCC2, or NKCC2 is highly expressed in kidney. That's why it worked as a water pill, and also work on the NKCC1, which is highly expressed in the brain. But it was not quite good, IC50, around 200 to 600 nanomole. In recent years, there are potential use for treating neonatal seizures, although the data are mixed because this drug doesn't get high enough to the brain for those diseases. So we look at this and then we see, okay, let's test this drug in 2 different AD mouse models and in iPS cells and also in the real world data. I'll first share with you the test in age E4 knocking mice. So for this E4 age knocking mice, special female mice at age over 16 months, in this case, we use 24 months old, really old female E4 knocking mice, a test in the so-called water maze test. So you put mice in the water tank filled with opaque warm water, and then the mice will use the cues to locate for the hidden submerged platform. So you train the mice for 5 days. This is the learning curve. You can see the solid white line is E3. They learned quickly during a 2 or 3 day training, but the solid red line, E4 knocking mice at this age is clearly impaired. They slow down the learning. And strikingly, 2 months treatment at 0.2 mg per kilo, which equals about a 1 mg pill human take daily. 2 months later, you will see this a dramatic improvement for learning, almost reach the E3 mouse level. And then we look at this functional effect of the bumetanide. So one phenomenon we know in this AG4 mice, if you record for this neuronal excitability, when you increase stimulation intensity, you see the response much stronger for the E4 knocking mice at this age, so-called hyperexcitability. As you can appreciate, bumetanide treatment for 2 months completely rescue this hyperactivity. And also when we test the long-term potentiation, which is a molecular foundation for memory formation, you can appreciate the E3A, the brown color had quite good LTP, but E4 had dramatically impaired LTP. 2 months treatment with bumetanide, complete rescue as shown on the quantification for the last 10 minutes. So, and then we look at the gene expression by single nucleotide-seq. We identify totally 18 clusters. And then if you look, the gene expression, 12 out of the 16 cell clusters have these E44 specific Alzheimer's disease genes reversed to a quite dramatic extent by bumetanide treatment, including all excited neurons, some inhibitor neurons, as highlight 4 subclass neurons here. So you can see this heat map indicating this upregulated gene in the E44AD brains. Many of the genes went down, and some downregulated gene in the AD brains went up after bumetanide treatment. So it seems this drug works in this age E4 knocking mice. And then we went on to test the drug in this E4 mice overexpressing also this FAD associated APP mutation. We use the J20 line cross with this E4 knocking line. For these mice, by about 10 months, this mice start to develop a beta accumulation. So we start treat these mice with bumetanide at 10 months and treatment for 13 months, still at 0.2 mg per kilo. After treatment, we took down mice, measured the A beta accumulation. You see on the right, there is a dramatic decrease in A beta accumulation based on quantification. It's about 50% decrease A beta load in both cortex and hippocampus. If you measure again this hyperactivity, in this case, both E3, E4 mice with APP overexpression had increased neuron excitability. Strikingly, 3 months bimetalin treatment bring down only in the E4 mice, but not in E3 mice, and also rescue E4 cause LTP impairment. So this tells us this drug may also work in these mice with both E4 and mutant APP expression. So how about the gene expression? So we did, again, the single nuclear seq analysis. Among those 25 cell classes, 6 of them were showing this significant reverse of this E4 for AD signature genes, including, again, the major excited neurons. And striking in this case, we start to see those gene reversion in both microglia and astrocyte. We did not see in this HE4 mice because in those mice, there were no gliosis observed. But in this APP model, clearly there were quite dramatic gliosis. Now we start to see the beneficial effect of this drug also on the glial cells. And then we look at this bimetalin effect on iPS cell-derived neurons. As you can appreciate, after treatment for 6 hours, you see this E44 uniquely increased gene expression went down, and this downregulated gene in the E44 AD brains went up for many of them after bimetalin treatment in human cells. You can probably appreciate this human neuron seems to get better reversion compared to the mouse neurons. So if we put all this bimetalin effect on gene expression in G20 E4 mouse, in the IPL cells or HE4 mouse brains, compared with the human E44 AD brains, you see the 3 major pathway overlap. That's the GABAergic synaptic pathway and circadian intraminal pathway. Many of you know those 2 paths have been recognized as a pathway get severely impaired in Alzheimer's disease and also morphine addiction. So we think bimetalin might act through these 2 pathway rescue to help on this. So finally, I want to share with you this so-called real-world data. So in collaboration with Marina's lab and also with Ben lab in Mount Sinai in the East Coast, we analyzed 2 independent so-called electronic health record database. So about 1.3 million individuals in UCSF and also about almost 4 million in Mount Sinai. So we filter through those 2 databases, so pull out those people who took this bimetalin and age over 65, because that's the time Alzheimer's disease usually start, and then control for some drugs also used for hypertension and edema. Finally, we get roughly about 1,200 in each database, and then we randomly match 10 randomly select a control group at a 1-to-2 ratio with similar drug treatment for edema, hypertension, to see whether bimetalin has any effect on Alzheimer's disease prevalence. As you can see in the UCSF database, the bimetalin treatment group compared to all 10 randomly select control group, there is a significant decrease in AD prevalence. And in the Mount Sinai group, there's even much more dramatic, 75% decrease in the Alzheimer's disease prevalence in this real world data. This strongly suggests that bimetalin probably has effectiveness in preventing or also potentially treating those Alzheimer's disease. So finally, in summary conclusion, I hope I convince you our experimental and real-world data evidence supporting this computational repurposing of bimetalin for ApoE4 related Alzheimer's disease. This paper was recently came out in this Nature Aging. We are glad, as a cover story. So now I'm planning for 5 or 6 different medical centers to try to start clinical trial to test bimetalin for AD patient. So finally, I would like to thank people who contributed to this work. I mentioned Alice, talent post student, has been leading this project. These projects are truly collaboration across 4 different labs. My lab leading this study in collaboration with Marina and UCSF, Ben in Mount Sinai, contributing on this real-world data, and Ben Chen on some analysis tools for this drug repurposing, the support from NIH for this funding. Thank you for your attention. I'll be happy to take any questions now.
Thanks so much, Yadong. We're open for questions. I guess the reason to do the clinical trial is that people could have something else underlying that causes them to take bumetanide that is associated with Alzheimer's disease rather than the taking that by itself.
So you mean the real world data, right?
Yeah.
Or
yeah.
the real clinical trial you're talking about?
Well, the real world, I'm just saying, I think it'll be important to do the clinical trial to see if this can actually reverse or delay Alzheimer's symptoms. But the association seems really
right. So you're right. Actually, I get a lot of call from those patients about whether they should start. So I always advise, because this is a water pill, is because it's a diuretic. For those aged people, I always suggest talk with their doctor to make sure they are safe on this drug because it can cause some dehydration. So
but it might be like people have some other cause that causes them to take this water pill, and that other cause is the thing that is connected to Alzheimer's disease, and it has nothing to do with taking the water pill.
Yeah, that's really possible. That's why when we did this real world data, we try to control the same drug those people take potentially on that, and also control for the disease severity, for example, hypertension, all others. So we try our best to do, but still possible. But I should mention, actually, recent, there's another paper came out just a week or 2 weeks ago, again in Nature Aging, from another group in, they use a very similar approach, but very different database, some proteomic, transcriptomic, a much bigger database, multiple database. They did a very similar approach, pull out top 66 drugs they think may be workable for AD. They focus on this Viagra, so you probably hear that news. So they see that drugs potentially protect AD in their real world database, which is in American, those are the insurance company database. It's even bigger. It's about 7 to 8 million, and quite interesting. And I'm glad to see in their 66 database top drugs, bumetanide was also included. It's very interesting. Bumetanide has an almost identical prediction score as this Viagra. So, but they focus on that drug. So now I'm collaborating with Dr. Jen, try to see whether in their database, in real world database, whether bumetanide could also work. So the final answer, I believe, I agree, will be the real clinical trial to test.
That's great. And then I don't mean to hog all the questions, but I'm just so intrigued by this. Is there any way to find in your real world data ApoE status and see if there's a greater effect?
That's not easy, actually. So that was the one caveat we discussed in our paper. The real world, the animal study all match the ApoE genotype, but the real world data is not genotype, ApoE genotype matched. So so that tells maybe this drug may even work for some E3 AD because it's not ApoE genotype stratified. Because as I showed for E3 individuals, the score still minus. So it's still indicating probably work at least to some extent on that. So now I'm working with Marina at UCSF to see whether we can gather more ApoE genotype information for some subgroup in this real world data.
Got it. Okay, plenty of questions. Oh, Professor Liu, do you want to ask your question? Liu Bai.
Yeah, very nice talk, Yadong.
Hey,
It's always great to hear your science. Very exciting. We're doing something similar on the AOS, and basically it's iPS-derived neurons doing looking at the phenotype and transcriptions, and also looking at animal models and patients data derived from patients. We have observed something. I want to know if you observed something similar as well. That is the iPS cells data, the gene expression profiles, oftentimes is a bit remote from data from patients, while the animal data is closer. That is, if you want to use what we call profiling as a phenotype to monitor drug effects, iPS is the best because you can apply drugs into the animal, you can apply many and dose response and so forth. It's much easier than doing it in animal. But if the data is not really reflecting the human situation, the human patient situation, because iPS-derived neurons are usually in the early stage of development rather than, aged brain in Alzheimer patients. Do you see something like that in terms of comparing gene profiling?
This is a very important point. We see some case. In general, I completely agree with you. If you use iPS, especially for aging-related study, there is some difference there because this iPS cell-derived neurons, like you said, just a few weeks in differentiation may not fully reflect the aging. But there is a uniqueness for E4 effect. The E4 effect in neurons, we believe, is because neuron, in response to challenge, culture itself, differentiate itself is challenge. So neuron will turn on ApoE during this culture. They start to trigger those E4 effect, which is a kind of advantage to the E4 study. But if you look at other study, it may not be true for this. So in the E4 case, we see usually the human iPS-derived neuron works actually so good, at least to the level like the knocking mice. In some cases, especially when you separate them into different cluster of cells, even better on that. But I would put a caveat on this, like I said, this is it's a very unique situation for E4 because for example, in knocking mouse brains, not all neuron has ApoE expression highly there because that's just a normal condition. But when you culture the iPS neuron in vitro, they all turn on ApoE. That pushes the E4 effect so dramatic.
Great, thanks. We should follow up discussion and leave times to others.
Okay, so AI put a quote in the chat. Are the compound treated transcriptomics obtained from mice, brain, or cell line, and is this database available?
Yes, all the database we did were uploaded to the GEO. So if you read the paper at the end for the data availability, you see a link or you see the number, you can get all this download.
Great. Evan Snyder, do you want to ask your question?
Sure. It's just a question that comes up whenever one wants to repurpose a drug for something that it was not initially intended. And I was just wondering whether you had given some thought to what might happen to try Bumex, which is a very powerful diuretic, on patients who are not hypertensive, don't have edema, don't have cardiac failure, and what might be the unintended effects when you do your clinical trial, as I know you have to do, and how often will you have to monitor electrolytes, weight, blood pressure, fluid status on these patients?
Yeah, that's a great question that have been extensively discussed, with clinicians in different medical center. That's the one major thing when we plan for the clinical trial. So yes, this drug will be used in the trial for those people they probably don't have hypertension, edema. So there's 3 pills in clinic, 0.5 mg, 1 and 2 mg. So like I mentioned, this 0.2 mg per kilo in mice, if you translate it in a way, is equals to about 1 pill per day in humans. I did not present, we have recent data in preparation for publication. We lowered tenfold of this drug in our animal models. We still see not as strong effect as this, high dose 0.2, but 0.02 for behavior, for semi-like physiology, we still see effect. So we are talking with the clinician to see we should start 0.5 mg pill, maybe even half of that. So it's just on discussion now. It should be monitored for all this drug effect periodically. I should mention, I mentioned in my talk, there was some ongoing clinical trials for autism and others, and people use 1 mg per day for 10 months. So they see some effect in the brain, but not related to Alzheimer's disease, but there are no clear significant side effect on those. So we hope we can figure out a dose and to use in the AD trial.
Thanks. It sounds like you've given it a lot of thought.
Thank you.
Okay, Dongfeng, do you want to ask your question? Dongfeng Zhang. Okay, maybe while
Dr. Huang, very nice talk. Yeah.
Well,
I'd like to ask a quick question. How about the CAMP score using your mice and IPS data rather than the data from the drug database?
Oh, yeah, that's very important question. We did calculate those. When we use this knocking mouse in vivo data, because it's a different cell cluster, you see this score show different. Some cells, especially for example, excited neurons, it's a very good score. But when you measure in other cell types, the score is bad. So it's from bad score to really good score. And some score better than this original CMAP database with this human cancer cell line. For the IPSS also depend on, because all IPS, when you test, we have the majority excited neuron, some inhibitor neuron, some other astrocyte. It's actually, again, excited neuron get the best CMAP score, which a little better than the original CMAP database.
Got it. Thank you.
Okay, Bin Xu.
Hi, very interesting talk, Dr. Huan. Congratulations finding this very interesting molecule. So I was just wondering, looks like from your talk, mostly talk about APP or ApoE for based pathology. Mm-hmm. Have you looked at others like such as tau-based neuropathology, like in mouse model? And another question I have is, how many like repurposed drug have you looked at that? Based on what you're finding, which you're finding a very interesting one.
Yeah, the first question, yes, the study is ongoing on this human E4 cross with PS19, this mutant human tau model. In our test, we always include E4 because the drug was originally pulled out based on ApoE4 genotype. So now we are testing E4 together with tau. And for how many drugs we tested, the very original was 1,300 drug. After that, we test another few more. So totally about 200 drugs now we test. 2,000 drugs.
Thank you.
Okay, Sumihiro Maeda.
Hi, Yadong. Nice to see you again. And thank you for very nice talk. And I have a similar question with previous person, but you have already answered part of it. But have you checked tau expression level in your drug treating mice? Because the effect of drug pretty similar with tau lowering. So I wonder tau may be lower.
You mean the tau levels?
Yeah.
Yeah, in our APP model, we did not measure tau because usually there's not much tau pathology. Like I just mentioned, we are working on 3 months treatment in E4 plus mutant tau model. So in that model, we'll focus on the tau measurement on that.
I see. Actually, I have another question. You mentioned that hyperexcitability in ApoE4 knocking mice, but I believe in Nature Communication paper you published, ApoE4 for iPSC neurons does not show hyperexcitability, right? At least spontaneous activity.
So if you look at this iPSL in our published work, we haven't done many iPSL work for electrophysiology, individual cell recording on that. So we actually don't know whether in this E4 or E3 iPSL derived neuron, E4 iPSL have hyperactivity or not. We don't know that. So now we have some ongoing study from a student project try to deal with that. But we did, instead of doing in vitro, we did so-called in vivo chimeric model. So we transplanted E3/4 human iPSL neuron into mouse brains and then give 6 or 7 months and then do the recording. So we don't know the answer now.
Okay, thank you.
Okay, Zhao Xuan.
Hi, very nice work. So I'm wondering, have you tried other drugs with a similar working mechanism as that drug, like the sodium potassium transporter? Do the other drugs also have effect on Alzheimer's?
We haven't tested many drugs. So we extensively test bumetanide. We test another drug also belong to this kind of a similar water pill. Not to this detailed extent, but we didn't see significant effect. So what we think, it's not sure to me, this is why this drug works is because this drug is NKCC1 NKCC2 inhibitor. I don't know. So there's quite a likelihood, probably why this drug works. It may not be because that. It could be because any unknown function of this drug.
So in your presentation, you said that you looked at the real world data for exposure to the drug. What does that mean? Like maybe they took it for a week 20 years ago, or do they have to take it every day?
So we did not restrict how long, but we cut, the people must use this drug at least for half a year, not every day, but at least they may stop in the middle and then they restart, but the total duration must be about over half a year.
For the half year before you got the data, or is it any half year period of their life?
So any half year. So we try to say if you split, after 65 or before, but when we do that, our N number was down quite dramatically. Total, we have about 1,200. If we further split, the power goes down. That's why I'm now working with Dr. Zhang in their database. They have about 8 million people for the insurance company record. So I hope we can get more people who took this drug, and then we can get more further detail separation.
Got it. Jay, perhaps the last question. Do you want to ask it?
Yeah, thanks for the nice talk. Maybe I missed this, but did you look at non-DE gene changes upon bumetanide? Considering the broad effect of the drug, I wonder a larger set of genes undergo changes in the expression upon this drug, regardless of ApoE4 effect.
Yes, when we look at this, when we try to match E4/4 to this drug, or E4/3 or E3/3, either of these 3 AD group, you see this drug significantly reach a statistically significant reverse the gene expression. But E4/4 reached the best. It's about minus 0.24, those kind of score. But if you go to E4/3, it's down to minus 1.9, minus 0.19, and then go to E3/3 AD, is down to minus 0.17, something like that. So it's a preferentially help on E4/4, but could also affect on 4/3 and 3/3. That means overall Alzheimer's disease.
All right. Thanks so much, Yadong. Really exciting. Just the ability to do this now it's amazing. And thanks so much for sharing this exciting work.
Thank you.
Thanks, everyone.
Thank you, everyone.
Yep. Thanks, Huan. Thanks, Yadong. See everyone.